Hello everyone, so today we are going to learn about oxidative phosphorylation, where we are going to produce ATP by using energy released from redox reaction. So we have two components involved. The blue part here is the electron transport chain, and also the green part here is the chemiosmosis that consists of ATP synthase. So first, we are going to look at our electron carriers that are formed previously from cellular respiration. So, NADH plus H plus and also FADH2 will travel from matrix of mitochondria to inner membrane of mitochondria. First, we are going to look at NADH plus H plus. NADH plus H plus as electron carrier, where it will donate 2 electrons to NADH dehydrogenase. NADH dehydrogenase will be reduced, and NADH plus H plus will oxidize to form NAD plus and also 2 hydrogen ions. These 2 hydrogen ions will remain in the matrix of mitochondria, and NAD plus will be recycled in the cellular respiration. 2 electron then is transferred to CoQ, where the CoQ is reduced and NADH dehydrogenase is oxidized. The transferation of 2 electrons to CoQ from NADH dehydrogenase will release energy, and the energy is used to pump hydrogen ion from matrix of mitochondria to intermembrane space. These 2 electrons will be carried by CoQ, and it will be transferred to cytochrome C reductase. Cytochrome C reductase will be reduced by accepting 2 electrons, and these 2 electrons will be transferred to cytochrome C. The transferation of 2 electrons from cytochrome C reductase to cytochrome C will release another energy, and it will be used by cytochrome C reductase to pump hydrogen ion from matrix of mitochondria to intermembrane space. These 2 electrons carried by cytochrome C transferred to cytochrome C oxidase. Cytochrome C will be oxidized, and cytochrome C oxidase will accept 2 electrons, and these 2 electrons will combine with the last electron acceptor, which is oxygen. The transferation of 2 electrons will release another energy, and it will be used by cytochrome C oxidase to pump hydrogen ion from matrix of mitochondria to intermembrane space. These 2 electrons and also oxygen will combine with another 2 hydrogen ions to form water. Now we are going to look at our second electron carrier, which is FADH2 that comes from matrix of mitochondria. FADH2 will donate its 2 electrons to succinate dehydrogenase, and it will oxidize to form FAD and also 2 hydrogen ions. FAD will be recycled in the cellular respiration process, where the 2 hydrogen ions will remain in the matrix of mitochondria. The 2 electrons will then be transferred to CoQ, and these 2 electrons will be carried by CoQ to cytochrome C reductase. These 2 electrons will be then transferred from cytochrome C reductase to cytochrome C. The transferation of these 2 electrons will release another energy, and the energy will be used by cytochrome C reductase to pump hydrogen ion from matrix of mitochondria to intermembrane space. Then, these 2 electrons will be carried by cytochrome C to cytochrome C oxidase. Cytochrome C oxidase will reduce, and these 2 electrons will be transferred to last electron acceptor, which is oxygen. The transferation of 2 electrons will then release another energy, and this energy will be used by cytochrome C oxidase to pump hydrogen ion from matrix of mitochondria to intermembrane space. The 2 electrons and also the oxygen will then react with 2 hydrogen ions to form water. The hydrogen ions, or the protons, that's been pumped from matrix of mitochondria to intermembrane space will causing high concentration of hydrogen ions in the intermembrane space that producing proton gradient between matrix of mitochondria and also the intermembrane space. This proton gradient will cause proton motive force, where the proton motive force will cause the hydrogen ion from intermembrane space to flow back through ATP synthase to the matrix of mitochondria. The flow back of hydrogen ions from intermembrane space to matrix of mitochondria through ATP synthase is an exergonic process. The exergonic process releases energy that drives the phosphorylation of ADP with inorganic phosphate to form ATP.